Chemical sensing with graphene, a quantum field theory perspective.
Thank you. Can you see my screen?
Yes, doctor.
All right, thanks a lot. Just 1 second, please. All right. So good afternoon, and many thanks to the organizers for the opportunity to present this work, which is part of a recent theoretical work within my group to study the optical conductivity in graphene and to propose a theoretical or a principle for chemical sensing, as I will discuss afterwards. So what is optical conductivity? So optical conductivity is the linear response transfer coefficient that characterizes the electric current triggered in a material due to the incidence of a frequency-dependent electromagnetic field. In general, optical conductivity is a tensor, so it is a two-indexed object, since it connects two vectors, which is the current and the electric field. So this is one of the earliest and most famous experiments on the optical conductivity and transparency in single and multi-layer graphene that shows a very good agreement between light or optical transmittance and simple Dirac fermion theory. Moreover, from a simple Dirac theory, as was already predicted in this experimental work, one can show that the transparency is proportional to 1 minus pi times alpha, where this coefficient pi alpha is 2.3%, in excellent agreement with experiments. This is compatible with a universal limit for the AC or optical conductivity, which is the square of the electric charge over 4 times the Planck's constant. So apparently, since this universal value is predicted from a simple Dirac fermion theory, that means a linear dispersion approximation for the band structure of graphene near the Dirac points, then it seems that no deviations or corrections beyond this model might be necessary to capture the behavior of this property. But then the question is really that all? I mean, if one looks at the atomistic structure of graphene, which is well described by a tight banding Hamiltonian, which includes both nearest neighbors hopping parameter, T in this notation, and also a second nearest neighbors coupling parameter, T prime. If one goes from this tight banding Hamiltonian to the explicit solution for the band structure, of course, we know this famous band structure in graphene, where we find the Dirac points, which are nearly linear in dispersion. However, the exact spectrum is more complicated, and there is indeed a contribution from the second to nearest neighbors hopping parameter, which predicts a small, however not necessarily negligible, quadratic contribution to the overall dispersion near each Dirac point. So the first coefficient is, of course, the Fermi velocity that depends only on the nearest neighbors coupling, but the second coefficient, the one that accompanies this quadratic term, can be interpreted within this group. This effective mass parameter has a range of values depending on how you estimate T prime. Since T prime has some uncertainty in the existing literature in between experimental and ab initio calculations, it has been reported on the range between 0.056 to 2.56 electron volts. So that gives a range for this effective mass parameter that, as I said, should introduce some corrections to this linear dispersion. And then the first question is how this small correction would in principle affect or not the optical conductivity in graphene. So this is the first question. And then a second related question is it possible to to use this effect for the purpose of chemical sensing, in the sense that is the optical conductivity sensitive to the presence of adsorbed dipole molecules in the surface of graphene? So let's first explore the first question. So we use for this purpose a quantum field theory formalism. So why quantum field theory? Quantum field theory is a powerful theoretical tool that allows to explore low energy continuum models to describe the physics of condensed matter systems. It allows for a rigorous analysis of continuous and discrete symmetries, and moreover, those analytical models can be equipped with parameters that can be calculated from ab initio calculations. So what kind of predictions can one do with quantum field theory? One can analyze many body interactions, strong electronic relations, such as Kondo effects in quantum dots and molecules. One can model topological effects and disorder. In particular, beyond disorder, also finite temperature effects, which are really, I would say impossible to predict only from ab initio calculations. And finally, which is more relevant to the present work, one can also involve the behavior of a given system in the context of transport phenomena in the presence of external fields. So we formulate a quantum field theory through a Lagrangian, which is the way it is usually formulated. That involves the contribution of this quadratic dispersion via an effective mass parameter that is captured in this theta parameter in the Lagrangian, which is proportional to the graphene Fermi velocity and also to this effective mass parameter that I was describing at the beginning. The equations of motion, that means the equation for the corresponding wave function arising from this theory, looks like a hybrid in between Dirac and Schrödinger equation, which is more or less expectable since we have a combination of linear plus a quadratic dispersion correction due to the second nearest neighbors coupling. So this model is implemented within the linear response Kubo formalism to calculate the tensor that couples the current to the external electromagnetic field. So this tensor, as I said, has two components. Within the Kubo linear response formalism is given by the current correlators calculated in the field theory. It is customary to choose what is called the temporal gauge for the electric field that gives a simpler proportionality factor in between this vector potential A and the electric field of the electromagnetic wave. And then in Fourier space, we define this polarization tensor coefficient, which is the Fourier transform of the linear response correlator in the Kubo formula. And then finally, the optical conductivity is calculated from this polarization tensor. So this is the main object to be obtained from this quantum field theory formalism. Technically, it is calculated from combinations of Green's functions. There are two important elements in these Green's functions. One is the so-called time order Green's function, or Feynman, and the other one is called the retarder in advanced Green's function. And from appropriate combinations of these two Green's functions via these momentum integrals, and these vertices, which are obtained from the field theory, we can calculate the different components of the polarization tensor, and hence from them to extract the value corresponding to the optical conductivity. So from the Kubo formula again, the optical conductivity tensor is given by the polarization tensor over the frequency, and there is a factor of 4 given by the valley degeneracy, because in graphene we have 2 Dirac points, that means 2 Dirac valleys, and the 2 spin degrees of freedom. So it gives a total of 4 times degeneracy. What we obtain after calculating these different integral terms is that the tensor is actually diagonal, which is expectable since the material is is isotropic at the mesoscopic level. And the diagonal terms reproduce the universal value that was quoted in the first experiment. To some extent, this seems surprising because we involved the second order or the second nearest neighbor's contribution in the model. And despite of that, we still recover, when we work at 0 temperature and 0 chemical potential, we still recover the universal value for the optical conductivity. However, there is a physical interpretation to understand this first preliminary result, because each of these polarization terms or components represent particle Hall excitations, as it is depicted in this diagram. If we go at a pictorial view of how these particle Hall excitations behave near the Fermi level, when the Fermi level, that means the chemical potential, is right at the direct point, that means the graphene layer is perfectly neutral, then the particle Hall excitations will be very close to this point, and then they will only perceive, these states will only perceive the linear dispersion. Since the curvature effects, which are captured by this inverse effective mass, will arise at slightly higher energy values. Then the question is, what happens if one considers a finite chemical potential? That means a charged material. The Fermi level is either above or below the direct point. In that case, the particle Hall excitations should start to perceive the effect of this curvature. And indeed, this is what happens. If we extend our calculation to involve finite temperature and finite chemical potential, instead of getting this simple constant universal value, we obtain a curve that is given by this sigmoidal shape. So the sigmoidal shape is connected to the universal value, since the asymptotic value that it attains is the sigma naught universal value that I was predicting. However, at finite temperatures, the slope of this sigmoidal curve starts to get steeper and steeper as you reduce the temperature until at 0 temperature you achieve this simple step, which is consistent with this universal result. On the other hand, the center of the sigmoidal curve is given by the argument in this formula, by the argument of these hyperbolic tangents, which is essentially twice the chemical potential in the frequency domain, plus a small correction. This small correction in our model is inversely proportional to this effective mass parameter. So in that sense, we corrected previous theories which did not involve this second to nearest neighbor's contribution, to show that there is indeed an effect of this quadratic dispersion in the optical conductivity. And the effect is displayed in a small shift of the center of the sigmoidal curve, which is proportional to this quantity. So how much is the shift? The shift is from the center at twice the chemical potential is proportional. If I go back to the definition of what is the inverse mass, is proportional to the second nearest neighbor's hopping. Okay, so the next question I wanted to answer is, once we figured out this correction to the optical conductivity, is it possible that the optical conductivity is sensitive to the presence of polar molecules? Maybe a small concentration of polar molecules in gas phase, as in this picture, which are absorbed at the graphene surface. And if this is so, this could provide, in principle, a way to for chemical sensing using the optical conductivity as a sensing variable. So in a simplified model, we assume that we have a graphene layer, and there is an environment of polar molecules represented as dipoles with a dipole moment, vector dipole moment P, which are randomly distributed in the surface. And the center of mass of these molecules is depicted by these capital R vectors, Ri, Rj. So we have an ensemble of these molecules with a given surface concentration. Again, we can use the tools, analytical tools of quantum field theory to describe how the presence of these dipoles will affect the optical conductivity. The way to do that, again, applying the formulas for the polarization tensor I was quoting before, we need to correct the Green's functions by the presence of these molecules. And the equation that allows us to connect these dipole molecules with the Green's functions is through what we call a self-energy in the context of many body physics or quantum field theory. So it's an integral of the average dipole potential we have. It is proportional indeed to the concentration of adsorbed molecules, and this self-energy will correct via this Dyson equation will correct the corresponding Green's function. So once we perform this calculation, it's a very involved calculation, but it can be done. We obtain remarkably a formula that looks exactly as the one we had in the previous case, but what we observe is that the parameters involved, that now you may see have a tilde on top. These tilted parameters are renormalized parameters, in the sense that they involve the dependency of the concentration of dipole molecules. They also depend, of course, on the molecular parameters, particularly on the dipole moment, P. So one can plot and represent the dependency of these different parameters in terms of the Bayer parameters as a function of the dipole concentration. So they clearly, the most sensitive one is actually effective mass. This curve was plotted for a particular case of ammonia molecules. And in terms of the optical conductivity, again, this is normalized by the universal value E squared over 4h bar. We see that these sigmoidal curves, the center of the sigmoidal curve gets shifted as a function of the dipole concentration. Actually, this shift, we can predict it analytically. It's given by this simple expression that depends on the renormalized parameters I was mentioning in the previous slide. So in principle, one can connect an experimental measurement of transparency where one could detect this shift in the center of the sigmoidal via these formulas with the dipole, with the molecular surface concentration. And this connection is interestingly not temperature dependent. It only depends on the dipole concept and on the molecular concentration to this ND parameter. So to conclude, we have studied the effect of this second nearest neighbors hopping correction in the dispersion of graphene, in particular monolayer graphene, both at 0 and finite temperatures. We obtained the exact, within the theory, of course, the exact analytical expression involving these corrections that opens the question of the possibility of chemical sensing by exploiting the sensitivity of the predicted optical conductivity on the presence of adsorbed polar molecules at the surface of graphene. Thank you very much. And if there is any questions, I will be happy to answer.
Thank you so much for the wonderful speech, Doctor. The stage is open for questions now. Once again, thank you so much for your speech, Doctor.
Okay, thank you very much.
The next invited speech is from Dr. Luca Di Giacomo, Italy, on the title Protein Corona of Nanoparticles for the Early Detection and Staging of Pancreatic Cancer.
Yes, thank you. Thank you for the invitation. Can you hear me?
Yeah, yes, Doctor. Please start
Okay.
your presentation.
Okay. So I'll talk about the possible employment of nanoparticles in nanomedicine, and in particular for the development of in vitro diagnostic tests for the detection of cancers, in particular for the pancreatic cancers. So as we know, nanoparticles are small objects, so whose size ranges from a few nanometers to a few hundreds, and they are characterized by manifold features like the size, shape, surface charge, chemistry, the composition, the presence of possible cargos, and all these features represent their so-called synthetic identity. But for nanomedical and biological purposes, we should take into account that when nanoparticles are exposed to biological fluids, as an instance in blood or blood plasma or serum, they interact with the environments. And as an effect, a biomolecular layer is formed on the surface of nanoparticles. This layer is commonly referred to as protein corona, because this biomolecular layer is mainly made of proteins. There is an inner layer, which is the so-called hard corona, which is made by high affinity proteins with the nanoparticle, and a soft corona, which is populated by low affinity proteins or weakly bound proteins. In our lab, we've been studying the possible biological effects of the protein corona in biological processes. Some of them are listed in this slide. So the state effects of nanoparticle uptake by cells, especially by immune cells, so the interaction with target cells for cancer therapy. And this involves also the mechanism at a cellular and subcellular levels. And in parallel, we being studied also which factors are responsible to the formation of the protein corona, as the surface functionalization, the role of the protein source, the role of the incubation or the fluid dynamic condition of incubation. In this context, one key aspect is represented by this statement. So the personalized protein corona. In 2014, it was demonstrated that protein coronas obtained by incubated nanoparticles with human plasma from different samples, from different subjects, are different. So in that work, the group led by Professor Morteza Mahmoudi used graphene oxide as nano platform and let human plasma interact with graphene oxide nanosheet. Human plasma obtained by blood collection from people suffering different pathological conditions. And they demonstrated by mass spectrometry experiments that the protein corona profile, so the composition of the protein corona was different, was subject dependent. So the personalized protein corona is a key factor for nanomedical science, especially for personalized precision nanomedicine. From the possibility to evaluate the physiological condition of a subject by an in vitro diagnostic test, also a personalized treatment can be designed. So this context is what is generally called precision medicine or precision nanomedicine. The general workflow for the different works exploiting nanoparticle protein interaction for diagnostic tests is summarized in this slide. So basically, we have a collection of blood and isolation of blood plasma from different classes of donors. Then the identification of a library of nanoparticles. In this case, we have living nanoparticles. These are liposomes with different surface charge, so positive surface charge, neutral or anionic surface. The interaction with plasma proteins so that a protein corona is formed. And the identification mainly by mass spectrometry experiments, which are the plasma proteins that bound, that absorb on the nanoparticle surfaces. Then by data analysis, we were able to identify which are the main proteins which detect the different pathological conditions. So the potential of the protein corona technology is disruptive, but today is very expensive because mainly the tools for protein corona analysis are based on nanoliquid chromatography, mass spectrometry, so very accurate procedure, but hardly translatable to the clinics. In brief, they do not fit the general guidelines for World Health Organization, according to which for cancer screening, the procedures should be also, of course, safe, effective, but also affordable and user-friendly. So we aim to use a different approach for protein corona analysis. Basically, the general workflow is quite similar. So a blood collection, the interaction with different kinds of nanomaterials. And in our works, we use a library made of metal nanoparticles, such as gold nanoparticles or silver nanoparticles, lipid nanoparticles, liposome of different composition and surface properties, or two-dimensional materials like a graphene oxide that interact with the human plasma from different donors, analyze the protein corona by SDS page, which is a technique which is commonly used to obtain a molecular weight distribution of the samples. So after protein corona isolation by centrifugation, we load the samples into a gel and an electric field is applied, and basically what it looks like a barcode is obtained. And after an image processing analysis, each pixel, the intensity of each pixel is recorded and evaluated as a two-dimensional function. The projection of the function along an orthogonal plane give a one-dimensional profile, which is the molecular weight distribution of the sample. And this is our readout for the analysis, and each profile corresponds to a single subject. We focused on the pancreatic cancer, pancreatic ductal adenocarcinoma, because unfortunately this malignancy is particularly aggressive, difficult to treat, and it is often related to dismal prognosis. And the reason is that often the pancreatic cancer is diagnosed when it is too late. So the survival rates are very low, and there are no today so effective technique for the diagnosis. So I'll show some of our main results. The first are obtained using gold nanoparticles with controlled size, it's around 120 nanometers, anionic zeta potential, and known concentration. We incubated gold nanoparticles with plasma obtained from a control group, so healthy donors, and pancreatic cancer patients. The molecular weight distribution of the corona proteins, the proteins populating the corona, are represented in these profiles. These are two representative profiles. So main difference are located within the low molecular weight ranges, 10 to 20, until 45, about 45 kilodalton. Then by applying statistical tools like the multivariate distribution in two-dimensional parameter space or three-dimensional, so coupling the information arose in the integral area in the low and intermediate molecular weight ranges, we obtained the possibility to classify healthy donors and pancreatic cancer patients with an accuracy of about 80%. By a variant of this approach, and by using graphene oxide as nano platform of reference, we were able to detect higher differences between the two classes, and they were mainly located between 20 and 30 kilodalton. Of course, the profiles are different because the nanomaterials in this case is another one. So before we had a specific profiles with some definitive peak. Here we have another molecular weight distribution. And in this range, we had the main differences between the two classes. The final outcomes for this test was very promising because we were able to classify correctly the healthy donors and the pancreatic cancer groups with an accuracy of about 94%. So we tried to go further and to employ a different technique, which is even more direct, as it's not need to isolate the corona proteins by centrifugation. The technique is a magnetic levitation. Magnetic levitation device is made by two strong permanent magnets with the poles of the same type facing each other. When a diamagnetic object is injected in a cuvette filled with a paramagnetic solution, it levitates until it reaches an equilibrium position. The equilibrium position depends on the experimental setup, so the magnetic field applied, the distance between the magnets, but depends also on the sample, particularly by the sample density. So the approach, the starting approach can be modified by evaluating the protein corona characterization with a magnetic device, a magnetic levitation device. We had some preliminary results that are summarized in this slide. The behavior of graphene oxide plasma proteins in the cuvette filled with the paramagnetic medium is this one. So there is a dense cloud which precipitates in about 5 to 10 minutes, and a residual population that reaches an equilibrium position that is the levitating population fraction. And this levitating fraction is more evident, more manifest for the pancreatic cancer groups. These are two representative samples, but statistically, the differences between the two classes were significant, and the test in a data set made of 30 samples reached a global accuracy of 93%. Of course, we had to enlarge the data sets to search for confirmation of these outcomes, but in general, we can conclude that we explored the potential of the protein corona technology to develop a non-invasive and effective tool for the early detection of pancreatic cancer. The corona profiles obtained by gel electrophoresis depended on the nanomaterials and the experimental condition. And in some cases, we obtained very promising results, reaching a classification ability of about 92%. And further studies will aim to enlarge the sample size and to employ complementary technique to achieve an effective translation of the protein corona technology in the clinical practice. And that's it. I would like to thank you all for your kind attention.
Thank you so much for the presentation, Doctor.
Thank you.
The stage is open for questions. We assume there are no questions for you as of now.